Thank you. I'm not a morning person, so I appreciate more you showing up at the last day of conference so early after the exciting party yesterday. Thank you very much. As I was introduced, I'm not a clinician. I'm a bioinformatician, actually nuclear physicist by the background. And Judith made a great introduction to most common therapies that we link to the germline findings in tumors. And I have to declare that I'm going to, I try to focus on the germline findings in patients with different tumors, but I will also sprinkle some somatic findings during the talk. Yeah, nothing to declare except I am scientific director in Genomics England, and just a quick introduction to the Genomics England to put talk into the context. So Genomics England is a company. We are owned by the UK Department of Health. We were established in 2013 to deliver whole genome sequencing into the healthcare for patients with cancer and rare disease. We close partners with NHS England, of course. And here are the settings of the Genomics England. So first I will talk about this clinical cycle. So this is kind of, probably many of you are familiar with. Patients get recruited, consented, DNA is extracted. We're doing whole genome sequencing at Illumina at the moment. We're also exploring Oxford Nanopore. Genomic data is analyzed, partially interpreted, and then clinical scientists at genomic lab is finish an interpretation and present patient data to the Genomic Tumor Advisory Board with expectation that will make contribution to the patient management. But this is not the end of the story. Data will flow into our secure research environment, where it gets merged with the secondary sources of the clinical data, and then all this data is accessible to the public, to the pharma companies for analysis, discovery, translational research, development of the new targets. And please, unless you already did, you are very welcome to join our research network. You can just search for the research network at Genomics England, get access to this many thousands of the whole genomes annotated with clinical data. This is what is in the research environment so far for the cancer patients. This is part of the 100,000 genome project that we completed in 2019, I think. But yeah, it took us a lot of time to publish paper covering this data set. It's appeared in January in Nature Medicine. It's shortlisted now by the European Society of Cancer Research for the highlights of the year. So unless you've already seen the paper, please take a look. Majority of whole genomes come from breast, colorectal cancer, kidney and sarcoma, lung, but we also collected a lot of these rare cancers. And as I said, it's linked to a lot of clinical data. We know patient diagnosis, staging, all hospital episodes, treatment response, survival, and cause of death. So again, welcome to our research environment. It's free, and compute is also free to run new discoveries. Okay, so I explained what Genomics England is doing, and now to the overview of the talk. So as we know, cancer is a disease of the genome, so I will go through the different rearrangements that we see in patient tumors. When I will zoom a bit into the homologous recombination deficiency in breast tumors, and also last couple of slides are recent reports on the clinical benefits of genomic sequencing for cancer patients. So I will use this graphical representation of the tumor genome through my talk. It's very important to understand what are these different tracks mean. So the two outmost showing tumor coverage and then normalized tumor coverage. So we derive data on copy number aberrations based on this coverage data. Then the next two tracks, this green one and this red one, are the densities of the single nucleotide variants and also indels. And then in the middle, you can see the structural rearrangement. Like, for example, these green arches are translocations between different chromosomes. So this sample was submitted as liposarcoma, but after sequencing, we realized that there is a very little tumor there. It's almost like a normal tissue. That's why it's so quiet. Okay, this is another sample of high-grade ovarian carcinoma, and this has become much more busy. And I just wanted to demonstrate here the case of biallelic loss of p53. And in this case, it's not direct target for treatment, but this is confirmed patient diagnosis, which of course is also very important. And this is biallelic because we have these missense events in p53, which also overlap by the loss of second allele. And it's very easy to see, to pick it up from the circus plot. You can see that the P arm of chromosome 17 have a loss of coverage, loss of one of the alleles, and the second allele have mutation, small variant. So this is an example of another type of rearrangements that we see in the tumors. This is a fusion of ATF1, AWSR1 in clear cell carcinoma. And again, we know that this is this confirmed patient diagnosis. Usually when we see this specific type of rearrangement, we see it in clear cell sarcomas. Yeah, here things get a bit busier and probably more interested. This is an example of osteosarcoma, and actually I learned a lot about this type of rearrangements on Saturday morning sessions on structural variants. So yeah, sometimes it's really work to wake up early and learn something new. So the first thing we picked up, you can see that on chromosomes 3, 7, and 12, there is a gain of coverage here. So like this MDM2, very well-known anchor genes for sarcoma, has 44 copies of the functional MDM2 gene. And we know that sarcomas with amplification of MDM2 is known therapeutic target. There are quite a few inhibitors on the approved on the market. But you probably also can see this number of green arches which connect these three regions. And also shown with green arches, and there are also these purple arches which show inversions when the pieces of the chromosome get inverted. So this is a likely indicator of the chromatripsis. Chromatripsis is when part of the chromosome gets fragmented, shattered, reshuffled, and then glued together in the random order. But here it looks like it was not just a single region of the chromosome. There were chromosomal regions from three different locations which get fragmented, shattered, glued together. So if you think what you have after this gluing of the pieces, some of these breakpoints, these junctions, they dropped in the middle of the genes. And I put these gene names around this circus plot. So then these pieces from the different chromosomes get together. You frequently end up with this hybrid protein. For example, in this case, piece of the MDM2 get together with the piece of this ROB1 gene. And sometimes it's also accompanied by the slippage of the frame. So you end up with completely new junction and also with completely new protein sequence. And then, as Judy described very nicely, these new peptides, they get at the surface of the cell, and they can be recognized by our immune system. So potentially, these cases can be very sensitive to the treatment with immunotherapy, with the checkpoint inhibitor, and also potentially with this personalized RNA cancer vaccines that still in the stage of trials. Very interesting case. Oops, I think I skipped one. Another case of endometrioid adenocarcinoma, and Judy described all the details this mismatch repair machinery deficiency, but I also want to talk about this polymerase epsilon complex and deficiencies in this specific gene. So then polymerase slides through the DNA. Polymerase epsilon has very important endonuclease activity. If the wrong nucleotide get recruited, then polymerase epsilon exonuclease activity can remove it and replace it with the correct nucleotide. So we know that polymerase E can have mutations that prevent this exonuclease activity. So we routinely looking at the germline of the pol-ER genes. And you know, here is a very interesting case when we did see stop gains in the pol-E gene in the germline. But this mutation was annotated as has conflicting interpretation and only one star. So it was a questionable case. It's like there is no obvious indication that this is a pathogenic mutation, because usually what we see is this missense mutation in the DNA binding domain. And DNA binding domain is at the N-terminus of the protein, and you can see that this stop gain happened in the middle of the protein. So it was very unsure whether it can be pathogenic variant or not. We also saw a couple of somatic variants, but again, missense variants, not many occurrences in the cosmic database. So it was not very obvious whether any of these variants are oncogenic or pathogenic. But again, if you look at this circus plot, you can see these green and red ribbons, which indicate very high mutational burden for both indels and SNVs. And if you look at the, like, what are the exact substitutions, what's the context upstream, downstream, you can see that majority of these variants are CA, yeah, blue one. And if you do, so we call it mutational signatures, like this prevalence of the specific types of SNVs or specific types of indels. We also start talking now about mutational signatures for copy number aberrations, for structural variants. But here is a simple case. I'm just looking at the SNVs. I see high prevalence of these C2As, and it recapitulates exactly the known signatures of this polymerase epsilon exonuclease activity domain mutation. So even though from our initial look at the specific variants in both germline and somatic, we were not sure whether the variants are pathogenic or oncogenic. If you look at the consequences of these mutations, you can get pretty confident that, yes, at least some of these three variants that I described on the previous slide are we say pathogenic for the germline variant and oncogenic for the somatic variant. So they do influence functionality, exonuclease activity of the polymerase epsilon. And this is, just to remind you, why do we care about these mutations? In a normal cell, there is a balance between tumor suppressor genes, oncogenes. Oncogenes are activate cell proliferation, and tumor suppressor genes obviously keep it down. So in amplification of the oncogene, like this MDM2 case that I showed you before, the oncogenes get more numbers, so the balance is disturbed. The same if you have activated mutations in the oncogenes, the balance between two is broken, and if you have deletion of tumor suppressor gene, then obviously, again, balance is on the side of the oncogene and proliferation. Okay, so we talk about types of mutations we are looking for. It can be small variants, can be structural variants, can be very complex structural variants. But it's also, as I shown you, very important to look at consequences of these mutations, like mutational signatures. Looking at the mutational signature at the tumor, at this footprint, you can infer what was the driving event that caused tumor to develop. Now, about setup of genomic testing in England. So as you seen from the name of my talk, we are doing both tumor and normal analysis. So we collect tumors from the sample, and we prefer fresh frozen tumors because formalin, which is routinely used by the pathology, is causing a lot of damage to the DNA. We now also introducing RNA later as a stabilizer for the DNA. And we also collect sample from blood, so we can compare these two with the reference genome. So if you find changes in germline, then this is inherited germline mutations. If changes only in the tumor, then we will classify it as a somatic mutation. And this is a setup of the genomic medicine services in our national health system. I will talk a bit about test directory because, yeah, I was asking about it before the lecture. So test directory define what are the routine tests that you're doing for patients with rare disease and cancer. Testing is done through 7 genomic lab hubs across England. Samples get collected, sent for the sequencing, variants are identified, annotated, interpreted, and then presented on Genomic Tumor Advisory Board. So this is what is test directory. And you can just Google to find the website. At the moment, they're just 2 Excel spreadsheets, one for the rare disease, one for tumor. But NHS is working on something more interactive. So it's a list of the genomic tests which are commissioned by the National Health System in England for cancer and rare disease. What's the technology that through which it will be delivered? And what's the exact clinical indication, what are the patients who are eligible? So it's covering a range of different techniques which are used in genomic testing for cancer patients. Whole genome sequencing is done only for the subset of tumors, and I will explain later how we selected them. This is just to explain what's one of the main strengths of the genome. If you go to the test directory, you will see that for some clinical indications, you have to do a number of tests. I think this is, this is an example of lung cancer. So it's this tiny biopsy from which you have to do immunohistochemistry, call different types of variants, also the pangenomic markers like tumor mutation burden, microsatellite instability, For hematological cancers, for sarcomas, we're doing a lot of FISH, cytogenetics for hematology, analyzed germline for the inherited variants, and also some pharmacogenomics. So with genome, you can just do it as a single test, and of course, this is a big advantage, also much easier to make interpretation of. Okay, so yeah, I described at the beginning this collection of cancers that we generated through the 100,000 Genome Project and the paper that we published as an outcome. So we used this data to make a decision about for which tumors we should be providing whole genome sequencing. So for example, if you look at the brain tumors, each column here is the target which is listed in the test directory. That means that it's either prognostic, diagnostic marker or target for approved therapy or for the clinical trial. And then, each row is a clinical indication, and then in each cell, we put prevalence of mutations in this gene for this clinical indication. So you can see that for brains, we have a lot of recurrent mutations which show up in the genes in the test directory in small variants and in copy number aberrations. For the variant tumors, 40% of patients will have homologous recombination deficiency, which we can infer from the whole genome sequencing, and also a lot of germline predisposition findings. Sarcoma is a type of tumor with very high number of recurrent structural variants and copy number changes. So those tumors with high prevalence of mutations, especially with pangenomic markers or with copy number change structural variants, were selected for whole genome, for providing whole genome testing as the standard of care within the genomic medicine services. And this is just to give you an example of a genomic marker for which we have now therapy which is approved. In this case, it's a PIK3CA inhibitor in breast cancers. But you can see that there is also very high prevalence of these mutations in different cancer types. So this is probably a target for future clinical trials. And now I will zoom into the homologous recombination deficient breast tumors. Yeah, luckily, Judith already gave a very good introduction to this, so I probably don't have to spend too much time on it. There is a complex of the proteins for the homologous recombination, and Judith told about BRCA or PALP2 have very high implication, and others deficiency in other genes probably have very different implication on overall disease. So deficiency in the homologous recombination makes it difficult to the repair of these double-strand breaks. Cells still manage, but it leaves these scars. I've shown them here in this lilac color. So you can look for the drivers of this homologous recombination, but you also can look for these scars. So if you look at the drivers, you're doing this BRCA1, BRCA2 targeted gene panels. But you can also look at the consequences of these deficiencies at these scars. So the panel which participated in the number of clinical trials for identifying patients with homologous recombination deficiency is this myriad my choice panel. It's only looking at the large rearrangements in the genome. We know that it's probably not very specific. It highlights some of the patients without homologous recombination deficiency as deficient. If you're looking at the whole genome sequencing, we can develop more complex and hopefully more accurate predictors of the homologous recombination deficiency that using different markers. And there are a couple of options available now. You can download these algorithms and implement them into the pipeline. We also did a lot of comparison between myriad panel and whole genome sequencing results. In addition to the large chromosomal rearrangements, it's also look at these signatures for the small variants, and very importantly for indels. These indels with microhomology is a very important marker of the homologous recombination deficiency. Judith already explained this concept of the synthetic lethality for the PARP inhibitor. If PARP is inhibited, then double-stranded breaks don't get repaired. They progress to the, single-stranded breaks don't get repaired. They progress to the double-strand breaks, and if cell is proficient in homologous recombination, it can repair these double-strand breaks. If cell have homologous recombination deficiency, then it will die. So this is how PARP works. And Judith also very nicely went through the details of the Olympia trial, but so I will just repeat that alaparib now is approved for the treatment of patients with BRCA mutations for the early breast cancers. And I just want to show you a couple of examples. Yeah. My favorite circus plot. So this is like a standard case, the BRCA case for the breast tumor with homologous recombination deficiency. So there is a germline variant in BRCA2, frame shift. It's already annotated on a clean bar as pathogenic with 3 star. And as was said before, we usually see biallelic inactivation. So the second allele is lost in this case. Very good. Patient is eligible for alaparib and hopefully will respond to it very nicely. Here, this case is a bit more tricky. There is no obvious germline BRCA variant. But you know, when I looked at the circus plot, there is a deletion of the BRCA. 1 copy is lost. So we started digging into the germline. And there are a number of in silico tools now that try to predict splice sites, gain or losses in the intron parts of the gene. So in this case, very interestingly, this intron variant was annotated with very high score. The highest is 1, so it's very close to 1, by the splice AI tool as a donor gain. So of course, we cannot base patient treatment on these in silico tools. We still need functional study, but this is a very good candidate for doing functional study and annotating this germline variant, whether it's pathogenic or not. And I just want to go through the couple of papers that will be published very soon that show benefits of the whole genome sequencing for cancer patients. Yeah, so the first paper should show up in a couple of weeks in Nature Medicine. It's a collaboration between Great Ormond Street. They were reviewing hematological cases, and Cambridge was reviewing pediatric cases with solid tumors. Altogether, almost 300 patients. And this is consecutive patients. This is not patients with high risk, or there were no any selection. It's just the patients that flow through genomic medicine services. You know, I forgot to mention that our whole genome sequencing take now between 2 and 3 weeks. So we can return results relatively quickly to make changes to the patient management. And you know, I copied this, this image. It's, of course, much larger image. Each column here is a patient, and there were almost 300 columns, so I only copied part of it. And at the top with green shown different non-WGS tests. FISH, copy number array, targeted DNA panel, targeted RNA panel, karyotype. And you can see that for these patients, in this case, it was acute lymphoblastic leukemia. There was a lot of testing done in parallel with the whole genome sequencing. And then at the bottom, these red squares are cases where whole genome sequencing had additional findings, which also made change to the management. So you can see that even though these patients were very broadly, extensively tested, there were still new findings from whole genome sequencing. So genomes here changed patient management in 7% of the cases, and interestingly, half of them, more than half of them, 11 cases, came from the germline findings. Even though patients were sent to the routine germline panels, it's usually panels analyzed only very limited set of genes which was relevant to the patient's disease. And in 11 cases, we had new findings when we looked at the germline in the whole genome. One of the examples was pathogenic PALP2 mutation in the patient with neuroblastoma. So when radiotherapy was applied, radiologist was asked to avoid breast areas on a patient because patients obviously have predisposition to the breast cancers. So it's not like the mutation therapy. It can be more subtle changes to the patient management. And in a third of these patients cohort, we had some additional disease-relevant findings in addition to what was derived from panels and FISH and copy number arrays. And some of these findings can be relevant if patients relapse in the future. And the second paper, very similar paper for the sarcoma cases, again, for the third of the patients, it's changed diagnosis. For a third of the patients, it's also identified targets for the personalized medicine, and also germline findings in 7% of the cases. I don't think it was expected for sarcoma patients. Okay, so here is the summary of what I presented so far. I haven't fit in time. So I told you about different types of rearrangements, both somatic and inherited, that can drive tumor development and predict response to treatment. I showed you some cases when mutational signatures or mutational scars can help you with interpretation of the point mutations that you picked up in either germline or somatic. And we start now gathering more and more evidence how whole genome is a powerful tool for making clinical decisions. Genomics in England now is more than 500 people, plus we have a lot of collaborations with research. We have a lot of collaborations with genomic labs, obviously, with NHS England. And of course, I have to acknowledge contribution of the patients and their families. In 95%, our patients give consent for the research. So as I said, all this data flow to the research environment for you to explore. And at the end, I just want to say that we are hiring. If you are talented bioinformatician with experience in cancer genomics and you want to change patient lives, then you're very welcome to join us. Thank you.
Thank you very much for this very insightful talk. Please feel free to ask questions, and also in the chat if you're following online. I'm pretty sure there will be one when we start with the microphone in the front.
Thank you for this presentation. My name is Katharina Wimmer. I'm from Innsbruck. I wanted to ask a very general question. When you do genomic sequencing, do you test also for copy number neutral LOH? Is that in this circular plots, in the circus plots
Yes,
represented?
Yes, definitely. There are three colors on this circus plot. It's either gain, loss, or loss of heterozygosity. So for both P53 and BRCA, we in both cases, I was showing loss of the second allele, but frequently we see loss of heterozygosity. So both alleles get mutated.
May I ask another question?
Sure, I think we have enough time, yeah.
Yeah. Regarding the case where you showed this truncating POL-E variant in the germline and the two somatic missense mutation, maybe I missed that. In the end, which one of these three did you make responsible for the signature that you saw?
I don't know.
My opinion on this is it's certainly not the germline variant. The germline variant may lead to loss of this allele, but it's probably the one that is in the polymerase region, and that is the 400-something variant. And this is important information. I see that in other papers too, that pathologists often think that truncating mutations in this gene are pathogenic, but we should not consider truncating mutations in a germline in POL-E causing an increased mutation rate. Thank you.
Yes, functional study would help the best. But yes, it was a bit suspicious that like we usually expect to see deleterious mutations in the DNA binding domain. So I agree with you that probably the one near position 400 was the closest.
The next question.
I think what is important, like if we consider patient management, that it's a high mutational burden, patient is likely to respond to the chemotherapy. Sorry, immunotherapy, checkpoint inhibitor.
Hello, thank you for the talk. My name is Dr. Peterson, I'm from Munich. I was wondering, for the first whole genome study on the pediatric patients, you showed additional findings. I was wondering whether there were some findings maybe missed in comparison to the standard techniques. When you apply all techniques together, did you also have cases where some fusions or SNVs or whatever were not picked up?
Yes, fusions, definitely. Mm-hmm. And also RNA panels, for example, don't pick up fusions with IGH enhancer. Yeah. So those were picked up by the whole genome. Yeah. But, you know, paper will be out in 2 weeks, so I didn't want to spoil it too much.
No, of course. Thank you.
Are there further questions? Then I would have another one. Do you have a rough estimation how many % of the targetable variants are in non-coding regions? Because I know some people also do exome sequencing in tumors. Do you have an idea how much the yield is growing when you're performing whole genome sequencing?
You know, some of the classical fusions are, well, they're not quite fusions when you translate gene under the control of the strong enhancer. You wouldn't pick up those from the exome sequencing, and you wouldn't pick up those from the RNA panels. So genomes are the most sensitive technique here. You know, we're learning more and more about fusions where, like, one of the partner can sit within the region of segmental duplication, which I don't think you would be able to pick up from the exomes. And then these more complex structural variant events like this chromathripsis reshuffling I was showing for the osteosarcoma. It's not something that can be picked up from exomes.
And maybe one more practical question. You showed a roadmap of how these analyses are organized, and I'm pretty sure you're trying to automate them as good as possible. What steps are the most difficult ones to automate in this process?
I would say final interpretation, which is done by the clinical scientists in England. I didn't mention it today. We have now guidelines for interpretation of somatic variants, because I think we were lacking it for a long time. So we try now to automate these guidelines as much as possible, but clinical scientists probably still have to do literature searches, assessments. But with the time, I think we building this database, interpretation knowledge base. So probably at some point we will just exhaust all and classify all the variants. And yeah, this will greatly improve our turnaround times, take less time for interpretation. But even now, if you think how many tests clinical scientists have to interpret, like DNA panel, RNA panel, probably some fish cytogenetics, it will accumulate to like 8 hours. If you can do whole genome sequencing within like a couple of hours interpretation, it will save a lot of time.
Thank you. Are there further questions?
I have also a question. So I saw that in one of the graphs in the first paper, you have DPYD, which is a germline variant. So most of the examples were directed to selecting treatments, but also these data can be used for to avoid toxicity. So, for example, did you see? Could you, especially for colorectal cancer or breast cancer, did you see patients in which did you change the therapy because of this germline?
Yeah. There is very detailed guidelines for DPYD that we implemented, first time I saw DPYD patients with both alleles mutated, it was so encouraging that you probably save patient life, avoiding toxic treatment. We also introducing now, there are a couple of genes for the leukemias, TPMT, not 51, I think, which also linked to the toxicity to the chemotherapies in leukemia. So this also will be soon in
I was also going to ask if you're planning to add more genes that we know they are relevant at the moment. So this is not a static list, like you're right, the dynamics, so you're increasing the number of genes.
Yes. Pipeline keep developing. It's not very easy to develop pipeline when you are in the clinical environment, and every change requires a lot of validation, extension to scope, a lot of paperwork to fill. But yeah, we're trying to keep up.
Thank you.
Thank you again, Alona, for a very inspiring talk, and I would like to close this session. I think we can agree it was a great idea to get up early after the party yesterday. Thank you to both speakers again, and also thank you for your attendance.